this is the fish preparation device. There is a culture chamber with cell inlet. There is a culture chamber is there with cell inlet and outlet. There is top, this one is the top part, and this one is the bottom part. Bottom part is having a membrane and perfusion mender and perfusion inlet and outlet. These are the inlet and outlets, and this is the bottom part, and this one is the membrane. So this is basically the fish preparation device. Here there is a culture chamber with inlet and outlet, and this one is the top part, and lower part is having the membrane along with the perfusion mender and perfusion inlet and outlet. This is the bottom part. So this figure we can see the basic structure of the fish preparation device. So this one shows the leakage, and this is the device. Basically this is seen as like this. Now what is the fluorescence in situ hybridization? We can visualize by this figure. A simple explanation. This one is probe labeled with fluorescent dye, and there is denaturation of the labeling with this one probe is labeled with the fluorescent dye. So there is coloring change, so simply to indicate that. And this is the DNA part. So this is denatured by application of the high temperature. So separated strands are there, and this is, I should take another color. So basically this blue part is DNA, and it is now hybridized. So application of the high temperature, these two strands are separated. Now suppose the probes are having a red color, so these two are also separated, and this will bind here like this simultaneously. So this will be the structure. This can be seen like this. So what are the different types of fish? So first one is ACM fish. This is alpha centomeric classical and midi satellite of fish. Catfish, this is cellular comportamental and analytist of temporal or cat activity by fish. Codfish, that is chromosomal orientation and direction fish. Deep fish, that is double fusion fish. And cryofish, that is cryosection fish. Locked nuclei analysis, LNA fish. Types of fish continued. So basically Q-fish is quantitative fish and has been mainly used for the measuring the number of telomeres. And repeats. Fusion single fish, that is FS fish techniques was initially devised for identification of the Philadelphia chromosome. Immune fish is a combination of two techniques. M-fish multiplex fish, a protocol of 24 coloring karyotype. Multilocus or ML fish. This fish assay was initially designed to screen for multiple microdeletions or syndromes in the patients. So how does fish work? So basically this is the patient's DNA, and it is by heating, that is we call, basically result in the denaturation and separation of the DNA strands. Both will come apart, and after that there is probe labeled with the fluorescence. So basically they hybridize on cooling several microorganism, that is target AC627BR, that is naphthalene. This one is contaminant, and naphthalene deoxygenase basically for the mRNA. RHLU basically astratrigine herbicides. Rhodococcus vrastalabins 16S RNA. KT1PHE trichoethylene Ralstonia euphorbia KT116S RNA. On what is the fluorescence microscopy? So basically light source is high pressure mercury vapor lamps, and tungsten halogen lamps or xenon lamps can be used. So basically filters are exciting filters to let a certain wavelength of light passes so that can excite the given fluorescence carried on the sample. Another one is the barrier filter to allow the visible light to so that the fluorescence can be seen by eyes or the image can be captured. So how are the data reported? Depending on the method, fish can be presented in two different ways. If fish is evaluated using a microscope and manual counting of labeled cell, the results are presented as cells per unit, liter or liquid or gram of solid analyzed. If fish is evaluated with advanced microscopy technique and digital image processing, the results are usually presented on a relative volume or area basis, which can be converted by laboratory to cell per unit or liquid or solid. So action of the fish, basically low resolution fish with metaphasic chromosome. Fish was originally used with metaphasic chromosomes. These chromosomes were prepared from nuclei that are undergone division are highly constant in each chromosome in a set, taking up a recognizable appearance. High resolution fish with extended chromosome and DNA fiber. The resolution of the fish is determined not only by the hybridization technique itself but by the nature of the chromosome preparation being studied. So what is the application of the fish? Basically detection of the numerical and structural chromosome abnormality, identification of the marker chromosome, monitoring of the effect of therapy, detection of early relapse or minimal residue, detection of gene deletion and gene amplification. What is the field of investigation? Fish-based assays have been developed within different fields of the investigation, including clinical genetics, toxicology, microecology, evolution biology, comparative genomics, cellular genomics, chromosomal biology. And what are the advantages of fish? Rapid detection and large number of cells can be stored in short period. Efficiency of hybridization and deletion is high, selectivity and specificity high. Cytogenic data can be obtained from non-dividing or thermally differentiated cell. Cytogenic data can be obtained from poor samples. A method can be adapted for automated systems. So what are the limitations of fish? Restriction to those abnormalities that can be detected with currently available probes. Only one or few abnormalities can be accessed simultaneously. Due to failure to detect signal, fish is higher sensitive for trisomy but less sensitive for detection chromosomal loss or deletion. Cytogenetic can be obtained only for the target chromosome. Thus fish is not a good screening tool for the cytogenic heterogeneous disease. Required fluorescence microscopy and image analysis. This is also important because it increases the cost of the detection method. And the recent research is there. Clonal evolution as detected by interface fish is associated with worse overall survival in population based analysis of patients with chronic lymphatic leukemia and